Hello, and this is our presentation for Lab 2, Measurements and Gel Electrophoresis by me, Daniel Marcinek, and Regine Canwell. The introduction for this lab mainly is what is gel electrophoresis. And gel electrophoresis is a method used by scientists that separates DNA or protein samples by charge and size. It can also separate DNA and RNA strands by length. What this method uses is an agarose gel, an electrophoresis casting tray, and micropipettes, which we'll all see later in this presentation. So more in depth, agarose is very porous, and it contains many different sized pores. Makes sense. The smaller the strand or particle, the farther it travels in the agarose gel. And scientists also use agarose to separate the positive and negative particles in a sample, which I stated before and we'll see later in the presentation. Another component of gel electrophoresis is trisacetate EDTA, which we'll abbreviate to TAE. It's a buffer that keeps a constant pH throughout the test and makes sure we have accurate results. Another component is the loading dye, which helps make the sample of your DNA sink to the bottom of the well. And then finally there's an electrical current, which causes particles to migrate to either positive or negative electrodes. Our hypothesis for this lab was that the samples with smaller molecules will move more quickly in the agarose gel than those who have larger molecules. Also, positively charged samples will move towards the negative end, and negatively charged samples will move towards the positive end. So the procedure for this lab is to first prepare the agarose gel. Next, you prepare the running buffer. Then you set up the casting tray. Next, you pour the gel into the casting tray. Then you add the TAE buffer to the casting tray. And then finally, you prepare the samples. I'll explain these better as we move along in the presentation. So the first thing to do is to prepare the agarose. So to do that, first we measured out 0.3 grams of the agarose powder. The reason we did this is because we needed our agarose gel. We're going to have 30 milliliters of agarose gel. And we want an agarose concentration of 1%. And 1% of 30 is 0.3. So as you can see here, we measured out 0.30 grams of agarose powder. Next, we added the agarose to a flask, which we'll use later. And then we started to prepare the running buffer. Now, since the TAE we used in the lab was a stock solution, it needed to be diluted. So to get 400 milliliters of a 1 times TAE solution, we needed 392 milliliters of water and 8 milliliters of 50 times TAE solution. The reason for this, well, to figure this out, you would do 50 times, that's the concentration, times X milliliters equals 400 times the 1 times concentration. So 50 times how many milliliters would give you 400? And that would be 50 times 8. So we need 8 milliliters of 50 times TAE. So preparing the next step in preparing the running buffer was we got 380 milliliters of deionized water. The reason we got 380 is because it would be more practical to round to the nearest tens, because this was a very large graduated cylinder, and it would be hard to measure out 392. So here we measured out 380, as you can see here. And then in a smaller graduated cylinder, we measured 12 milliliters of deionized water, so we would get 392 milliliters of deionized water, which is what we want. So here you can see 12. And next we measured out the 50 times TAE, that's the more concentrated solution. We needed 8 milliliters of that, so we used a large pipette to extract that. And here you can see we got 8 milliliters. So then this is our 400 milliliters of 1 times TAE. Then we went back to the agarose solution. So before microwaving the 30 milliliters of 1% agarose solution, this is what it looked like. Afterwards, after microwaving it for about 30 seconds, it became very hot, of course. And it cleared up like this, which is what was expected, which is what you want. Then we added after we let the agarose cool a little bit, but did not let it solidify, we added it to the electrophoresis tray. There it would solidify, and we added a comb. So as the gel solidified, it would create wells, which we'll see later. So preparing the samples, this is me adding 15 milliliters of water to each tube, which we would then add 5 milliliters of dye to. Since this is a longer video, I'll skip to a part where you can clearly see me adding water. I apologize for my slow computer. That is extracting 15 milliliters of water from a small vial, and then adding it to one of the vials. We would next add 5 milliliters of dye to, except for the 4 dye tube, which we would need a less amount of dye and more water because it was more concentrated. Okay. So that was me adding 15 milliliters of water to a sample. Next we have Regine adding 15 milliliters of water to one of the vials. Okay, moving on. So after we added our dyes to the water, we then micropipetted the dyes and the water mixed together and added them to the gel. Micropipettes, more specifically, are used to measure very small amounts of liquids, and they're very accurate, so they were very useful in getting an accurate measurement of how much liquid we needed. So this is me adding a sample to our electrophoresis tray. Okay, as you can see, I executed that perfectly. No, I'm just kidding. Okay, moving on. I'm going to stop this part here, and the rest will be in part 2 of this presentation.